Welcome to the Women's Health Seminar Series, which we will be running for, probably we'll do virtual for a while, even if we start opening up, just because it allows more people to attend our series, which I think is always a good thing. And before I get started, I want to recognize that we live, work, and play and participate in a community on unceded, ceded, and traditional territories of the 203 First Nations, along with the 38 Métis chartered communities, each of which possess their own unique traditions and history right here on this land that we now refer to as British Columbia. We acknowledge the importance of the Truth and Reconciliation Commission of Canada's call to action, the UN Declaration of Rights of Indigenous Peoples, and the BC Declaration of Rights of Indigenous Peoples Act. In all of our work, we're committed to ensuring Indigenous women's rights to health and safety and equal opportunity to participate in a manner that recognizes and respects Indigenous cultures and traditions. Today, I am joining you from Vancouver, which is part of the unceded homelands of the Coast Salish people and the traditional territories of the Musqueam, Squamish, and Tsleil-Waututh nations. So I should also just briefly say that the Women's Health Cluster, we've been going for about 2-3 years now. We have over 230 members. We acknowledge support from individual and provincial and federal sponsors, without which we couldn't run this series. So we encourage everyone to become a member, especially trainees. We do have some money to give out. Obviously, people aren't traveling anymore, but you can use it for registering for conferences. And we're exploring some other opportunities so that we can give a trainee some money, and you can't get the money unless you become a member. We have that small little caveat in there so we can make more of you members. It is a multidisciplinary event to celebrate, and I'm going to stop sharing so that Dr. Frick can share her screen, celebrate women's health and spotlight women's health, because there's not as much research done in this area as we would like. But Dr. Frick, I am very pleased to present to you. She's a distinguished professor of psychology at the University of Wisconsin-Milwaukee and the chief science officer of Estrogenics, I'm probably not pronouncing that properly, Therapeutics Incorporated. Dr. Frick has nearly 30 years of experience studying the neurobiological mechanisms underlying memory formation, with a focus on cellular and molecular mechanisms through which estrogens regulate memory consolidation. I don't know why I'm reading this, because I probably know that off by heart. She has published nearly 90 papers in leading neuroscience and neuroendocrinology journals. I can attest to that. She's also an editor of the book Estrogens and Memory. I have a copy. Actually, my microphone is right on top of it right now. Otherwise, I'd show it to you. I should have done that earlier. It's called Estrogens and Memory, Basic Research and Clinical Implications. It's a fantastic book published by Oxford University Press just last year. She's the recipient of the Society for Women's Health Research Medtronic Prize for scientific contributions to women's health, and the Alzheimer's Association Southeastern Wisconsin Investigator of the Year. Is that what WI is?
Yeah.
And then, as many of you will know, I like to give a fun fact, because it's great to hear all these accolades of these researchers that we have giving talks for us. But it's also nice to know that they are a real person as well. And of course they all are. And I have a lot of fun facts because I've known Karyn for quite a while. When we could travel, we've had quite some adventures. And one of them, I will say, is that we had quite an experience, and we've walked for miles and miles and kilometers finding a very special Italian chocolate shop in Torino, Italy, which was fantastic. I think it was something like 1736 or something. Anyway, it doesn't matter. Her daughter, as you may, or you probably don't know, but is also a famous baker in her own right, or burgeoning baker in her own right. She has a fabulous brownie recipe that we made again last night, Karyn. And, so my daughter and I were having our feast of brownies yesterday. Anyway, without further ado, I present to you Dr. Karyn Frick. Thank you.
Thank you so much, Lisa. Thank you very much for inviting me, for the Women's Health Cluster, for inviting me to give this seminar. I'm appreciative of all of you attending, especially those of you who are eating your lunch and watching at the same time. I wish I was in British Columbia giving this talk because it's a wonderful, wonderful city, but we'll have to do this virtually for now, given the times. So I'm going to focus today on our work in female animals, focus on sort of women's health and focus on women. So I do all of my work in mice as animals, as models of menopause, and just as you'll see, trying to get a better handle on how estrogens are regulating brain function. And our work is motivated largely by, if I can get my computer to advance itself, there we go, by this particular sex difference. So as you are hopefully aware, women are at greater risk of developing Alzheimer's disease. And this image from the Alzheimer's Association shows the estimated lifetime risk of developing Alzheimer's disease at ages 65, 75, and 85, with women in the gold and men in the purple. And you can see, obviously, women have a higher risk of developing the disease over their lifetime than men. And that's true even when you take into account the fact that women live longer. And we think that this is due, at least in part, to hormone changes during menopause. And as many of you are probably aware, hormone levels are quite high after puberty in women. We're talking about estrogen and progesterone levels. And then they decline precipitously in the early 40s, in the beginning of perimenopause. And then they're quite low from the 50s and beyond. And we think this is significant for the brain because estrogens, turns out, are very important trophic factors for neurons in the brain that mediate cognitive functions. So here we're talking mainly about neurons that live in the hippocampus and the prefrontal cortex. And so we think that when you take away these estrogens, you thereby deprive these neurons of an important trophic factor that keeps them healthy and functioning well. And we think that that increases their vulnerability to the detrimental effects of aging and Alzheimer's disease. So this would seem to be a relatively simple problem to solve. If if the loss of estrogens at menopause increases a woman's risk for Alzheimer's disease and dementia, then we can simply replace those hormones and decrease those risks. And you're probably well aware that things are not that simple when it comes to hormones. So while there are definitely beneficial effects of hormone therapy, particularly estrogen therapy, on bone health and cholesterol, and particularly for menopausal symptoms like hot flashes, there are reported side effects on things like breast cancer, uterine cancer, and blood clots. And so these purported risks have caused millions of women throughout the country, throughout the world, to terminate hormone therapy and have changed recommendations for hormone therapy use. And so, when those initial studies came out suggesting that there were risks of these detrimental side effects, we started to wonder, as well as others in the field, we realized we didn't know very much about how these hormones were affecting organs like the brain. And emerging evidence in the early 90s was suggesting that, in fact, these cognitive regions of the brain were very sensitive to estrogens. And so that has started us on a couple decade search these days for trying to figure out how estradiol or estrogens like estradiol, so this is 17-beta estradiol, it's the most potent of the estrogens. So how does estradiol regulate the function of regions of the brain like the hippocampus, which in this cartoon is in the warm colors here? It's a bilateral medial temporal lobe structure. So how does estradiol act on these brain regions to form a memory? And so today I'm going to focus on kind of three areas in which we're doing our work that are all sort of centered on this idea of how can we reduce memory dysfunction in older women. In the first, I'll spend most of my time talking about our work in trying to determine how estradiol regulates memory in young female mice, because young mice are an optimally functioning system. And so if we can understand what estradiol is doing in that system, we can then, that will help us figure out what's going wrong in aging and neurodegenerative disease. We then would like to know, what's going on? Why does hormone loss lead to memory dysfunction in models of menopause and AD? And then finally, I'll talk a little bit about our efforts through this company, Estrogenics, that Lisa mentioned, in terms of developing novel treatments for reducing memory decline in menopause. So I'll start by just mentioning that most of the work that I'll be describing today looks at the effects of estradiol on the hippocampus. So I've already shown you that cartoon about humans. This is another cartoon illustrating where the hippocampus is in the rodent brain. It is still a bilateral medial temporal lobe structure, but in rodents, it extends much more dorsally, as hopefully you can see my cursor here. But it serves very similar functions to humans. And in terms of estrogen receptors, we know that the two canonical intracellular receptors, ER alpha and ER beta, are located throughout the hippocampus. So in this, hopefully you can see my cursor. In this illustration, you can see ER alpha and ER beta throughout the dorsal to ventral extent of the hippocampus. And the size of the dots indicates the relative abundance of the receptor. And you can see that there are a lot more ER betas than there are ER alphas. There's a third estrogen receptor that I won't talk very much about. I'm not going to talk very much about estrogen receptors today, but there is a third estrogen receptor that's located in the hippocampus. That's called G protein coupled estrogen receptor. This is a traditional G protein. So it's a plasma membrane receptor that functions much like other G proteins. But this is also located throughout the hippocampus. All three of these receptors are also found in the prefrontal cortex as well. Now, this is an illustration of what has become the sort of classic effect of estradiol on the hippocampus. So the hippocampus was first determined to be very sensitive to estrogens back in the early 90s in the pioneering work of Bruce McEwen and colleagues, where they looked at a specific type of neuron in the hippocampus called the CA1 pyramidal neuron. CA1 is an area of the hippocampus, and these pyramidal neurons are excitatory. Their cell body looks like a pyramid, hence the name, and they have these very extensive dendritic processes, these apical processes and basal dendrites. And these dendrites have what are called spines along the dendrites. They're these little knobby outgrowths of the spine that are places where these neurons make connections, form synapses with other neurons. So the thinking in the field is that the more dendritic spines one has, the more connections with the neurons, greater synaptic plasticity, therefore better memory. And what was found back in the early 90s was that during the phase of the rodent cycle, so they don't have a menstrual cycle, they have what's called an estrous cycle that lasts about 4 to 5 days. In the phase of the estrous cycle where estrogen levels are high, we call that phase proestrus, there are a lot more of these dendritic spines than when estrogen levels are low in the adjacent phases of the cycle, particularly this phase called estrus. And you can see that difference quantified down here in the graph, where proestrus is green and estrus is blue. And you can see there's about 30% more spines in the proestrus female than in the estrus female. We can see similar effects of estradiol in animals who've had their ovaries removed. So these are bilateral, what we call ovarectomies. So you'll see that abbreviated as OVX throughout the slides. In animals who are OVX but receive vehicle, those animals have far fewer dendritic spines than animals who are overactomized but replaced with estradiol. And so you can see that in both dendritic spines and the synapses formed. And over the last couple of decades, we've come to know that the hippocampus, estradiol does a lot of other things in the hippocampus as well. I'll talk a bit about its effects on cell signaling, but it also has rapid effects on epigenetic processes like DNA methylation and histone acetylation. It affects gene transcription, neurogenesis. Lisa has done quite a bit of work on neurogenesis. It alters various forms of synaptic plasticity like LTP and multiple kinds of hippocampal memory. And as I said earlier, our work is predicated on this idea that the age-related loss of estrogens may exacerbate the effects of aging on memory. Oops. Okay. With the exception of the Alzheimer's mouse model that I'll discuss towards the end of the talk, all the work I'll be showing you today is from C57 Black 6 mice. These are just wild type mice in which we've shown that the onset of memory decline in females during aging is associated with the loss of hormone cycling. When the animals are when hormone levels are starting to decline in these animals, so is memory function. And we and others have shown that if you replace estrogens in these animals, either middle-aged or to a certain extent old animals, you can help to reverse some of this memory loss. And then I put this image here to remind me to tell you that with the exception of a couple of studies at the very end of the talk, where I'll note this, all of our estrogen treatments are delivered directly into the brain. So the animals are chronically cannulated into the dorsal hippocampus, the dorsal third ventricle, or the medial prefrontal cortex. Okay, oops. All right, so how do we measure memory in these animals? There are a lot of different ways in which we could do that. And there are a lot of ways in which we have done in the past, but we've been primarily focusing on 2 object-based tasks that are very rapidly learned. So we just use common household objects that are placed in a big empty white box. The animals are habituated to the empty white box, and then they're trained. And by trained, I mean they're simply placed in the box with 2 identical objects, and we give the animals 30 seconds to explore the objects. Well, the animals have to actually be in the box until they've explored the objects for 30 seconds. So in this case, it's all internally motivated by the animals. They're intrinsically interested in objects because they're something new that they've not seen before. And so that fosters their motivation to explore the objects. And then after a delay one important thing I wanted to mention is all of our treatments, with the exception of a few that I'll mention along the way, are given immediately after training. So we can pinpoint estradiol's effects to the memory consolidation phase of memory formation. We know that the effects of estradiol on the brain are very rapid, that happen within minutes, and that memories are consolidated in this task within about an hour or 2 after training. So giving our treatments immediately after training helps us to pinpoint effects on consolidation and effects on these rapid brain processes. So again, we train the animals, give them these post-training drug treatments, and then for the object recognition task, in this case, generally 48 hours later, the animals are placed in the box where now they're tested, and they're presented with an object that they've seen before and a novel object. Because mice are intrinsically motivated by novelty, they like new things. If they remember the familiar object, they're going to spend more time than chance, which in our case is 15 seconds, with the novel object. So this task allows us to test how well the animals remember what the objects are. We use a second task called object placement to get a sense of how well the animals remember where the objects are in space. So in this task, training is exactly the same. 2 identical objects, post-training treatments. The delay is a little bit shorter, but during testing, instead of introducing a new object, we simply take one of the old objects and move it to a new place in the box. So this allows us to, if the animals remember where the objects were, they should spend more time than chance exploring this familiar object in a new place. And so the key difference between the 2 tasks is simply what happens during testing. And what I will show in the next slide is an example of the kinds of data that we see in this task. So to orient you, because you'll see a lot of graphs like this, the animals can spend a total of 30 seconds with the 2 objects. So 15 seconds, so 30 seconds is the maximum amount of time they can spend with the objects. So 15 seconds is chance. And you see that animals who receive a hippocampal infusion of a vehicle spend chance amounts of time with the 2 objects. So they don't prefer one or the other, and that tells us they're not remembering that they've seen one before. On the other hand, animals that receive a bilateral infusion of estradiol into the dorsal hippocampus spend more time than chance with vehicle with the novel object, and less time with the familiar object, telling us that they do remember the training objects and they're preferring to spend time with the new object. And we see the same thing with the object placement task. And we've seen this kind of memory enhancement over and over again for the last 10 or 12 years. And so we use this behavioral effect as basically a behavioral probe to try to then understand what in the brain what are the important cellular molecular changes that cause that memory enhancement? So how does estradiol enhance memory? And there are a lot of ways in which it could do that, one of which is via what we call classical mechanisms. Estradiol is a lipid. It's synthesized from cholesterol, as are all the sex steroid hormones. So it very easily moves through lipid bilayer membranes, binds to ER alpha and ER beta inside the cell, and then that hormone receptor complex binds to an estrogen response element on the DNA and causes gene transcription. And while it's possible that that accounts for some of the more longer term effects of estradiol on memory, we don't think it can account for these rapid effects on consolidation, because that's thought to be a relatively slow process compared to non-classical effects, in which estradiol can trigger cell signaling within, just a few minutes, 5 minutes or so, and can also, rapidly alter epigenetic factors, gene transcription, and protein translation. So we think that estrogen receptors, including ER alpha and ER beta, are actually acting at the membrane to mediate some of these rapid effects. And in fact, as I mentioned, GPER is a membrane protein itself, so well poised to trigger some of these very rapid changes. And so in the next slide, this is where we are so far in terms of identifying the receptors in purple and blue, the cell signaling mechanisms in green, and the epigenetic and genetic factors in orange that we have so far shown are necessary for estradiol to enhance memory. I'm not going to go through this in exhaustive detail today. As Lisa mentioned, if you want to learn more, we have a chapter about this in the book that we put out last year around this time, that Lisa also has a chapter in, as well as I know a few of you in the audience do too. But this is a really excellent, if you're interested in estrogens and memory, it's an excellent resource that goes all the way from in vitro work, very reductionist, looking at synaptic plasticity, all the way up through clinical studies in humans with a variety of different species, including birds, rodents of various sorts, monkeys, and humans. So if you're interested in the topic, that's a good resource, I think. But I'm going to focus here today on kind of the center of this model. And, right. So back probably 15 years ago, we knew that estradiol regulated spines. We knew that estradiol regulated memory, but we didn't know exactly how. And so we started becoming interested in this particular cell signaling molecule called ERK, or extracellular signal regulating kinase, because this kinase gets phosphorylated by this upstream kinase, MEK. And when that happens, well, actually, phosphorylation of ERK is necessary for memory formation, regardless of estrogens. It's true in males and females. ERK phosphorylation is necessary for long-term memory formation in the hippocampus, the amygdala, and other brain regions. And back in the 80s and 90s, there was a literature also showing that estradiol could increase ERK phosphorylation very rapidly within just a few minutes. And so we started putting those two literatures together and thinking, if estradiol can increase ERK phosphorylation, and ERK phosphorylation is necessary for memory, is necessary for memory, then maybe ERK phosphorylation is necessary for estradiol to regulate memory. And so we did a couple of studies back in the late 2000s, one in young animals and another in middle-aged animals. And I'm showing data from the middle-aged study right here. We found that if we infused estradiol into the dorsal hippocampus or dorsal third ventricle, that it within 5 minutes increased phosphorylation of particularly the P42 isoform of ERK. This is the more important of the two isoforms here. Oops, didn't mean to do that. And if we infuse the animals with estradiol plus an inhibitor of ERK phosphorylation, it not only prevented estradiol from increasing ERK phosphorylation, it prevented estradiol from enhancing memory. And so this was really exciting for us, because this was the first time that anybody had shown that rapid activation of cell signaling was important for estradiol's effects on memory. And we, this is, I guess, all I'll say about estrogen receptors. In some later studies, we looked at the role of ER alpha and ER beta in mediating the effects of estradiol on ERK and memory. And we found that these guys, activation of IDA receptor did enhanced memory, and they did so via ERK. But interestingly, they did so in concert with metabolite glutamate receptor 1. In fact, these guys had to interact with mGluR1A in order to increase ERK and ERK phosphorylation and enhance memory. So this was interesting because it showed this activation of these receptors, not in the nucleus, but here at the membrane in conjunction with a glutamate receptor. We've also looked at activation of GPER, which at least in our hands for the kind of memory that we're looking at, well, activation of GPER does enhance memory, but again, in our hands, it doesn't seem to do so via ERK phosphorylation. It does so by activating a different MAP kinase called JUNK, or C-Jun N-terminal kinase, which phosphorylates cofilin, which leads to actin polymerization, which is necessary to make spines. And so we found that GPER activation, in fact, did increase spines in a paper that we published last year in the Journal of Neuroscience, and this is one of our lovely neurons. We found that GPER both increased spinogenesis in the hippocampus as well as enhanced memory. Okay, so that's all I'll say about receptors. What I wanted to talk a little bit more about is one of the downstream effects of ERK phosphorylation. ERK can do a lot of things, but one of the things that it will do is phosphorylate this protein called mTOR and start mTOR, or mammalian target of rapamycin, signaling. mTOR is important for local protein synthesis inside dendrites. And in order to make those spines that I showed you, you need proteins. And so we suspected that mTOR might be important. And so in this one study we did back in 2013, we found that if we infused estradiol alone into the dorsal hippocampus, we saw an increase in ERK phosphorylation as we had before, but that was blocked by inhibitors of ERK, this upstream kinase called PI3 kinase, and rapamycin, which is an inhibitor of mTOR signaling. And not only did those inhibitors block ERK phosphorylation, they also blocked memory. They prevented estradiol from enhancing memory. And so, that was suggestive of the idea that this local protein synthesis was important for spinogenesis and memory. And then a few years ago, we finally had the opportunity to look at whether these two signaling kinases were involved in estrogen's effects on spines. And so we got to ask the question about whether dorsal hippocampal activation of ERK or mTOR was actually necessary for estradiol to increase those spines. And these are just some images from our mice of those who were treated with vehicle or with estradiol. And so basically, so this is work we did in collaboration with Vicki Lean at Hunter College in New York City and Maya Frankfurt of Hofstra LIJ School of Medicine on Long Island in New York. And the work in my lab was done by a former graduate student, now Dr. Jennifer Tuscher. And Jen infused estradiol into the dorsal hippocampus and looked at the apical and basal dendrites of CA1 pyramidal neurons. And she found that within 30 minutes, there was a significant increase in both basal and apical spines, and that lasted for 2 hours. And that wasn't terrifically surprising or novel, but what was really interesting was that when she infused estradiol in combination with the ERK inhibitor or the mTOR inhibitor, it totally wiped out the effects of estradiol on spines. So this was very exciting because this was the first time that anybody had shown that this rapid activation of these cell signaling cascades in the hippocampus was necessary for estradiol to regulate dendritic spines. We'd known that estradiol regulated spines for a long time, but we didn't know exactly how that was happening. And so it would seem that these data support the idea that rapid activation of this local protein synthesis is important for these spine changes. And so that was interesting in and of itself, but what was also interesting and unexpected was that these infusions into the dorsal hippocampus also affected spines in the medial prefrontal cortex. So here again, we looked at apical and basal spines in the prefrontal cortex and found, this is the more sort of prelimbic, infralimbic area, that we saw an increase in basal spines within 2 hours in the prefrontal cortex, which was super unexpected to me because I was solely focused. This is a focal infusion directly into hippocampus. But I think certainly I tend to forget, the field tends to forget in many cases that these brain regions don't exist on an island unto themselves. They're all connected into circuits. And so if you affect the functioning of one part of the circuit, you might affect the others. And so in this case, we were seeing that not only did estradiol infusion in the hippocampus affect what was going on in terms of spines and prefrontal cortex, but if we blocked ERK and mTOR phosphorylation in the hippocampus, that also prevented estradiol from increasing spines in the prefrontal cortex. So that was really exciting because that suggested that these two brain regions are talking to each other and that affecting cell signaling in the dorsal hippocampus was preventing estradiol in that brain region from regulating spine density in another brain region. And so that caused us to wonder whether these two, whether an interaction between these two brain regions is actually necessary for estradiol to enhance memory. I mean, it may be that we infuse estradiol into the hippocampus of females and things happen in other brain regions, but they're sort of ancillary to the main, effects of estradiol on memory. We wanted to see whether there was a real causal interaction between these two brain regions. So Jen took this on as her thesis, her PhD thesis work. And we first had to do some work in the prefrontal cortex because we didn't know really anything about the prefrontal cortex's role in mediating memory in these two tasks. So she first had to establish a role for the prefrontal in these two tasks. And so she did that in two ways. First, with a gain of function experiment to see if estradiol infused into the prefrontal cortex would enhance memory in the object recognition and object placement tasks. So if we increased, boosted the function of the brain of this brain region, could we enhance memory? And then second, if we should do a loss of function experiment. So what happens if we inactivate that brain region? What effects might it have on memory? And then assuming those both worked out, and spoiler alert, they did, then we could look to see whether there was a specific interaction between the medial prefrontal cortex. If we inactivated the medial prefrontal cortex while we were infusing estradiol into the hippocampus, does the inactivation of the cortex prevent estradiol from enhancing memory? So do these two brain regions need to be active simultaneously in order for estradiol to enhance memory? And we thought the best way to do that was to inactivate one of them and see what happens. So for the gain of function experiment, Jen sunk cannula into the medial prefrontal cortex after recovery, trained the animals, and now immediately after training, the animals received an infusion of HBC, which is our vehicle, and or estradiol. And in both tasks, she saw that estradiol treatment enhanced memory in both object recognition and object placement. It increased spine density, as we had seen before with the dorsal hippocampal infusion. But interestingly, that interaction didn't go both ways. So we didn't see any effects on CA1 spines, at least at this 2-hour time point. So the gain of function study worked. What about loss of function? Here we decided to inactivate the medial prefrontal cortex using a chemogenetic technique called DREADS, which is designer receptors exclusively activated by designer drugs, which is basically a virus that in our case here was conjugated to a promoter for excitatory neurons, the CAM kinase 2 promoter. So we're specifically targeting those pyramidal neurons that also live in the prefrontal cortex. This virus carries with it a mutated muscarinic receptor that gets when we treat the animals with a synthetic ligand that binds to this mutated receptor. In our case, the ligand here we're using is clozapine N oxide, or CNO. When CNO binds to this mutated receptor, it hyperpolarizes the neurons and prevents them from firing. Now, this is a temporary lesion or temporary inactivation, if you will, because once CNO is metabolized, the neurons go back to functioning perfectly well. So it works works perfectly for our purposes because we can inactivate the brain region of interest immediately after training, like we would for any other pharmacological treatment, and within several hours, it's gone from the system and the neurons are functional again.
So here we infused the virus into the prefrontal cortex. Now we had to, again, these are overactimized female C7 black 6 mice. Here we have to wait 3 weeks for the virus to express itself in neurons. But then after that time period, here instead of, in this particular experiment, we're inactivating the brain, the prefrontal cortex with this IP injection of CNO, and then we test the animals. This is an example of our sort of representative example of our expression. This is the mutated, the virus, the HM40I virus. And then down here, EGFP virus is our control virus. So we have a control group that is a combination of mice receiving the control GFP virus, as well as sham operated animals who just got an injection of saline into the medial prefrontal cortex. And you can see that for both tasks, inactivating the medial prefrontal cortex impairs memory. Here we're testing animals at shorter time points than what I've shown you before, so that our control animals in fact do show memory in the task. So our gain of function worked, our loss of function worked. So now we knew that the prefrontal cortex was important for these two tasks. So now we could go and look at the interaction between the two brain regions. And so here, Jen put cannula for delivering estradiol into the hippocampus, and she infused virus into the medial prefrontal cortex. Waited 3 weeks, did training, and then immediately after training, the mice were injected with either vehicle or CNO intraperitoneally, and then infused with either our HPC vehicle or estradiol into the dorsal hippocampus. Now, this is a very busy slide, very busy data. Let me break it down for you. So it's object recognition and object placement. The white and gray bars are all the control groups. So those are animals in all 3 groups who are infused with HPC vehicle. None of them should show memory, and they didn't. The lighter purple bars here are the control animals, the sham and the GFP controls that are infused with estradiol, and they do show memory, as you would expect them to. The animals that didn't show memory were the animals infused with the virus and estradiol. So in this case, yes, it does seem like activation of the medial prefrontal cortex is actually necessary for this dorsal hippocampally infused estradiol to enhance memory, telling us that, yeah, these two brain regions must be activated simultaneously for estradiol in one brain region to enhance memory. So here's our updated model. So here's all the things I was showing you earlier that we think are going on in the dorsal hippocampus, but now we've got other brain regions that are playing a role. So medial prefrontal cortex, where we know that estradiol is important and ERK and mTOR is important for regulating things like memory and spines. Now, this is interesting because there is a direct connection between the dorsal hippocampus and the medial prefrontal cortex, but it's rather sparse. It's not a super heavy projection. In fact, the ventral hippocampus projects much more heavily to the medial prefrontal cortex. And so we and other people in the learning and memory field who have been looking at dorsal hippocampus, medial prefrontal interactions think that maybe other brain regions are involved, and that this one nucleus in the thalamus called the nucleus reunions may be playing an important role in mediating the communication between dorsal hippocampus and medial prefrontal cortex. So one of my grad students, my current grad students, Miranda Schwab, has been trying to inactivate the nucleus reunions to look to see what kind of role it may be playing in learning and memory. So if we're inactivating that using a different DREAD, a kappa opioid receptor mediated DREAD. But if we inactivate the nucleus reunions either about 10 minutes before training or immediately after training, we do see impairments in learning and memory, suggesting that these data are from the object placement task. So that does suggest that nucleus reunions is important for memory. And now she's working on doing some circuit-specific inactivations to see whether the specific connections between medial prefrontal cortex and reunions, or reunions in dorsal hippocampus, are playing a role in estrogens, or playing a role first in memory and then in estrogen's ability to enhance memory. I should also mention too that I've stipulated here, this is in females, I don't have time to talk about our work in males, but we've done some work with cell signaling in males. Estradiol enhances memory in males, but it seems to do so using cell signaling mechanisms that are independent of ERK. So that's an interesting, may have interesting implications for sort of drug development for men and women. Okay, I know I'm running kind of low on time, so let me try to get to quickly some of these other parts of our model. So we've talked a lot about how estradiol regulates memory in young mice of both sexes. So why does it do that? We've been looking at using an Alzheimer's mouse model to try to understand what happens to why women are at increased risk of things like Alzheimer's disease. The risk factors for late onset Alzheimer's disease include, of course, age and female sex, but also ApoE4 status. And just briefly, apolipoprotein E is a lipid-carrying protein that's involved in cholesterol metabolism and transport. It's made by astrocytes largely. And it transports the toxic A beta, particularly A beta 42, into cells for disposal. A beta is the main constituent of amyloid plaques that litter the brains of Alzheimer's patients. It also transports cholesterol into neurons for metabolism into sex steroid hormones like estradiol. We have each of us have 2 copies of one of 3 different alleles of ApoE. E2 is thought to be protective. E3 is neutral in terms of risk of the disease, and E4 increases your risk of the disease. E4 turns out to be least effective at transporting A beta into cells for disposal. And so maybe that's one of the reasons why people with 1 or 2 copies of E4 are at increased risk. People who are carriers of ApoE4 have more and earlier accumulation of amyloid plaques, phospho-tau, which leads to neurofibrillary tangles, and brain atrophy, as well as cognitive decline. And there's an association between ApoE4 genotype and women's risk of AD. The women who are double E4 carriers are at more risk of AD than men who are E4 carriers, or men or women of any other genotype combo. And interestingly, estrogen therapy is less beneficial to cognitive function in double E4 women than double E3 women. It's possible that the ability of ApoE4 to transport cholesterol and dispose of A beta may contribute to increased A beta risk in women by a number of different ways. Number one, by actually decreasing estradiol synthesis in the brain, because the estradiol is made in the brain and it's important for memory function there. It could decrease estradiol-induced neuroprotection, increase A beta and phospho-tau accumulation, and thereby promote synaptic and neuronal degeneration. So how do we mitigate this increased risk in women? Number one, we can use mouse models to try to figure out what's going on mechanistically. To, for instance, look and see why sex and ApoE genotype effects exist, why double ApoE4 women fail to respond to estrogen therapy. And hopefully that information will allow us to develop treatments to mitigate those effects of sex and ApoE4 in women and perhaps increase their responsivity. I'll just say very briefly that we've been using a what we call an Efab mouse model that's developed by my colleague Mary Jo Ledoux at the University of Illinois at Chicago, where these animals carry 5 familial Alzheimer's disease mutations, and specifically 3 APP and 2 presenilin 1 mutations, and then they overexpress human E2, 3, or 4. We've been working with the 3s and the 4s. The double E4 mice have impaired spatial memory, accelerated A beta accumulation. They say, as you can see here, more A beta 42 and oligomeric A beta in the cortex in the hippocampus. And you can see these by 4 months, but they're particularly striking at 6 months, which is the age at which we've been working with them. And we've found that the male and female E4 animals are impaired in the object recognition and object placement tasks. Actually, the female E3s are also somewhat impaired. Really, the only animals that could learn these tasks were the male E3s. We also have looked at dendritic spines in these animals. We've categorized them by shape, with the mushroom-shaped spines being considered the most mature, stubby, sort of intermediate, and thin being most immature. And we see, a genotype effect in terms of total spines, mushroom spines, and stubby spines, where the E4 animals, males and females, have significantly reduced numbers of total mushroom and stubby spines. That's reflected also, we've done some Western blots looking at the presynaptic protein, synaptophysin, and the postsynaptic protein, PSD95. Both of those are reduced, again, straight by genotype, so both males and females. Where we do see some interesting sex differences among the E4s is in gliofibrillary acidic protein, or GFAP, which is an astrocytic protein. So these data, this increase here suggests more astrogliosis or more inflammation in the brains of E4 females. They have less phospho-Krebs, the transcription factor Krebs, suggesting that perhaps they have reduced gene transcription. And interestingly, we haven't looked at ER beta in these animals. We have looked at alpha, and ER alpha levels are actually higher in male and female E4s, which is interesting. They're actually the lowest in the male E3s, who do the best in the memory tasks. Oops. And then finally, with respect to this model, I'll just say that just before, well, as COVID was hitting and the lab was shutting down, we finished behaviorally testing a group of animals that were, these are all females that are double E3s, E3-4s, and double E4s. And here these animals were cannulated. They received bilateral infusion of estradiol immediately post-training. And you can see that estradiol enhances memory in animals who have 1 or 2 copies of E3. But in animals who have 2 copies of E4, just they do not respond to estradiol in terms of either one of these tasks. And these data are very new. We're still counting these data, still actively doing the medial prefrontal cortex. So I have just a little bit of apical, CA1 apical spine density data for you. But so far the data suggests that estradiol is also increasing spines in the double E3s and the E3-4s, but not in the double E4s. So, you know, we're hoping we are still analyzing, counting spines and doing Western blots, trying to figure out why these E4s are not responding to estradiol. And we hope that that information will help us figure out why women who have 2 copies of E4 don't respond as well. And then I know I'm a little bit over time, but I just have 2 more data slides. I just want to show you where we're going in terms of this last box here, developing novel avenues for drug development. So I'm one of the co-founders of this company called Estrogenics Therapeutics, along with my collaborators, Dan Sam of Concordia University of Wisconsin and Bill Donaldson at Marquette University. And Bill's a synthetic chemist who has developed a library of ER beta specific agonists. And our lead compound so far, called EGX358, is 750-fold more selective for ER beta over ER alpha, which is about 20 times more than any other compound that's out there. Which you see here in red, that's EGX358. So you can see it superimposed over estradiol. And a few years ago, one of my former grad students, Jae-Kyun Kim, ran several studies where he showed that if he compared the effects on memory of EGX, multiple doses, to DPN, which is a commonly used ER beta agonist, as a positive control. He found that EGX enhanced memory in both object tasks. I'm just showing you one here. If we infused it into dorsal hippocampus, if we injected it intraperitoneally, or if we administered it via oral gavage. Again, this is an acute treatment done immediately after training. We've since done this was another, got this in by the skin of our COVID pants study, where my current grad student, Aaron Fleischer, now treated mice orally for 6 weeks and found again that the compound enhanced memory, but he also was looking at hot flashes. So here, these again are overectomized mice, but we're trying to induce, hot flashes are sudden increases in temperature. And so here we're trying to induce that sudden increase with an injection of a compound called Sanctide, which is a neurokinin 3B agonist. And we're measuring temperature by infrared recording of tails, so looking at tail temperature. So this animal is one at rest, and you can see that you can barely kind of see the tail. This animal's been injected with Sanctide. The animals release heat out of their tails, And so this animal is showing it, by when body heat is increased, you'll see an increased release of heat out the tails, and it becomes lights up red. And so we have this on the top here is showing you the effects of just a vehicle injection. You don't see much in the way of increase in body in tail temperature. But after an injection of Sanctide, you see a very rapid increase in tail temperature that's transient. And all three of our compounds, estradiol, DPN, and EGX, which is our novel compound, reduce that tail temperature. So we're very excited about that. This compound is still in some preclinical trials, but we are hoping to move into phase 1 clinical trials fairly soon. So just to sum up, we talked a lot about estradiol's effects in the brain of females. We're currently working on some other avenues here, looking at the extracellular matrix and protein degradation, and looking at the role of astrocytes in estradiol's effects on memory. I told you we're looking at nucleus reunions in circuitry. We haven't talked about any of our work in males, but there we're focusing on cell signaling and interaction with androgens. In our Efab model, we actually have funding from the University of Wisconsin system to test our novel compound EGX358 in this Efab model. So those mice should be arriving later in the spring for those studies. And then, as I said, we're in some more preclinical testing with EGX. So I'll end there, acknowledging the hard work of all of my students and postdocs and our collaborators, Mary Jo, Dan, and Bill, as well as our funding sources. And at that, I will end, and hopefully there's a few minutes for questions.
There are. Thank you so much. I encourage people to use their emojis to tell our post-COVID world to give a clap. It's very hard, as I'm sure many of you are very well aware, to give these Zoom talks. I mean, yes, we're very used to them now, but it's always nice to have some affirmation from the audience, and nobody can laugh at your jokes or
I know.
constructive. So it's nice to have those little emojis. So you feel free to, I have a couple questions, and I know there's some questions about, can you buy EGX358? That's from Elena and me. I'll put that there. And she also wants the brownie recipe, which I'll share with you later.
I also have a great chocolate chip cookie recipe. I'm happy to share with you too.
Perfect.
But yeah, no, I see. Yeah, hopefully people can buy EGX358 at some point.
Yeah. For millions of dollars.
But Dr. Colares, if you're interested in using it for research purposes, yeah, we could probably talk with Bill to see if he can synthesize some of that for you, and you guys can try it out in your system.
Oh, sorry.
And yeah, no, I'm just looking through the chat here. I see there's a question about
what delivery mechanisms
in clinical trials.
Yeah.
I mean, obviously we're hoping to for oral of administration for human clinical trials, which is why we've been using the oral formulations for EGX. So in fact, this new study that we're doing with the Alzheimer's mouse model, again, from an academic standpoint, where I want a brain mechanism, I mean, I'd love to be infusing it into the brain more, but, clinically that doesn't really work very well. So we're planning to go ahead there with either oral gavage or delivering it through a hydrogel the animals would eat. So I think the future studies will continue with the oral formulation, and knock on wood, hopefully that will keep working and they can get to clinic. But the really exciting thing about that compound is that it's, we'll probably be working with it, pitching it more for hot flash relief, because that's a primary symptom that lots of women are interested in having relief from hot flashes. But it has this really great sort of secondary benefit in terms of enhancing memory. So
well, and I think hot flash, although it's like a little messy, but the hot flash and night sweats research, it does look like it's also tied to cognitive issues as
absolutely
well, right? So
absolutely right. I mean, yes, there's certainly direct connections between cognition and vascular function there, but also hot flashes disrupt your sleep, and sleep is important for cognition. So there are multiple ways in which
roots. Yeah, exactly. I have a question about the, I know I look at the chat, I'll make sure I do a look, but so you see an increase in your alpha, right? In your E4, what are they, E4 fat mice, but yeah, estradiol isn't having beneficial effects, but like, or at least in the young mice, it does seem like it's working through ERA alpha. So any ideas there? Are you planning on following that up? I mean, it's obviously an obvious one would be, hey, it's not fun. They're increased, but they're not functional, or they're
exactly. That's a possibility. So I don't know. The student who's been doing that work, Lisa Taxier, she's looked at ER beta for that sex differences experiment. Sorry, ER alpha for the sex difference experiment. But so far, she's blotted for ER beta in the estrogen experiment. And there's actually no differences among the genotypes in ER beta levels, and there's no estrogen effects on ER beta. So as you said, it could be that there's ER beta are there, but they're simply not functional. But she hasn't yet blotted for ER alpha in that study. So I don't know whether that's going to mirror what we saw in the first study with the sex differences or not. So that's going to be a really important question. And certainly, we should get a little bit more information when we start infusing or treating that Alzheimer's model with the EGX compound, which is, specific for ER beta. We've got to do a lot more there. It's interesting that I don't really know how to explain the fact that the mice who did best in the memory tasks had the lowest ER alpha. I don't know. So
I think that's really, I just think that's an important point to point out too. Just because something is more doesn't mean that it's related to better function.
Yes, absolutely.
Alexandra Hiria has a question too. I don't know if she wants to unmute, or you want me to ask a question for you, Alexandra?
I'll give it a shot. Let's hope my 8-month-old doesn't chime in.
Totally fine. We got dogs and cats and all sorts.
Great talk, Karyn. I was curious if you had any thoughts on how hormonal contraceptives might affect any of these consolidation processes, either by reducing endogenous estradiol production and or via action of ethinyl estradiol?
Yeah, that's a really great question. My read of the literature on that is, we don't know nearly enough about what oral contraceptives might be doing for cognition in women. And the literature seems to be kind of messy. At least that's my interpretation of the literature on oral contraceptives. Usually those kinds of studies are comparing women who are on oral contraceptives to women who are naturally cycling. If there are differences seen among those groups, in my opinion, they tend to be very subtle, and there's really there aren't whopping treatment effects there in my experience. So I don't know that there's really any effect of either contraceptive use. I don't know that there's any necessarily considerable effects of contraceptive use in daily life. However, one really interesting question that I think the field hasn't addressed is, what are the effects of women who start oral contraceptive use very early, say around puberty? Because of a variety of medical problems, they go on oral contraceptives, but their brains are still developing, right, until they're in their early 20s. And, I don't think, that would be a very interesting question to understand, if going on artificial, exogenous hormones, whether that has any kind of long-term effects. I doubt it has much in the way of effects in terms of their normal functioning when they're young and in their 30s and 40s. But I wonder what impact that might have as those women reach menopause and beyond, whether there's any impact one way or the other of that very early exogenous hormone use. I don't know.
I will say, I think Elizabeth Hampson has some work on oral contraceptives and some forms of cognition. I don't know about memory consolidation in particular, and in humans, obviously not.
Yeah.
But yeah, I don't know how dramatic the effects are. Well, we are now at, I have more questions, but we are now at 1, and I'm trying to be a good host and keep us on time. But there's some more questions from Elena, but I know that Elena and you know each other, so you guys can chat. I thought it was...
I'll email you the answers to your questions.
And I tweeted a little bit. That's what I was looking down doing. But I tweeted your title about the Neural Village, which I really encourage people to read. Karyn has a really fantastic review also in Frontiers in Neuroendocrinology called something about like it takes a neural village or something. And so I really appreciate that part too. So if everyone could just join me in thanking Dr. Frick again for a fantastic talk. And thanks to all of you for joining us virtually. And I really do hope at some point we can meet
I know.
in person. So thank you so much.